Seeds are not passive objects. They are sophisticated biological sensors operating like a complex computer. They do not simply detect soil. Instead, they perform a rigorous integration of environmental data, effectively making a choice. The seed processes multiple inputs simultaneously. Only when all critical conditions are met does the seed shift its state. This triggers the switch from metabolic dormancy to active growth. The soil is merely the cradle that holds the seed. It is not the trigger for life. The journey of a seed begins not with growth, but with a physical trigger called imbibition. This is the master switch that ends dormancy. As water enters the dry seed, the internal tissues rehydrate. This physical swelling is the first essential step in reactivating the seed's metabolism. Once hydrated, the seed synthesizes gibberellin hormones. These chemical messengers act as the internal signal that growth processes must commence. Gibberellins trigger the breakdown of stored starches into energy. This chemical conversion provides the fuel required for the seedling to break through its protective coat. Without this process of water uptake and hormonal activation, the biological clock of the seed remains paused, keeping it safely tucked away from an environment that may not yet support life. Seeds act as light detectors. They use a protein system called phytochromes to measure the ratio of red to far red light, which indicates whether they are under a canopy or in the open. A low ratio of red to far red light signals the presence of a canopy overhead, which would shade the plant. Under these conditions, the seed remains dormant to avoid competing for scarce light. Conversely, a high ratio of red light indicates open, sun-filled ground. This tells the seed it has a clear path for photosynthesis, effectively triggering the germination process. These light signals are integrated into the seed's genetic decision-making pathway, acting as an environmental gateway for survival. Roots exhibit gravitropism, orienting themselves downward toward gravity. This is managed by statoliths, dense starch-filled organelles within the root cap cells. Shoots exhibit phototropism, responding to light signals to ensure the plant grows upward toward the sun for efficient photosynthesis. By integrating gravity and light, plants establish a clear directional path for survival, ensuring roots anchor the plant and shoots capture sunlight. Many assume soil is a biological requirement for germination, but it's actually just a support medium. It serves primarily as a reservoir for water, oxygen, and structural anchorage. When we isolate these variables, hydration, temperature, and gas exchange, we find that seeds germinate just as effectively on damp paper towels or in sterile hydroponic grids. By removing the variables of soil pathogens and competition, we can create ideal conditions. In controlled agricultural environments, germination rates consistently exceed 99%. Ultimately, soil is merely the cradle. If you provide the necessary environmental signals artificially, the seed remains entirely indifferent to the medium underneath it.